Why is it important? Well, we've talked about that, so I'm going to skip over this slide. Basically, because we can be immune matched to large segments of the population, we take away one of the major issues that normally would affect stem cell transplant. And I'd like to get into the details of that a little bit further. The next slide is a chart. It looks a little bit like we've been obsessed with a deck of playing cards in part, but it helps understand what's significant about homozygous parthenoid stem cells. If you look at the first part of the chart, if you have each donor and each recipient have what are called HLA factors or HLA loci, and there are 3 or 4 of these that are critically important in any transplant program. If you can match 3 or 4 so that there is no alien antigen present, then you have a good transplant, and you can perhaps use a very light dose of immunosuppressants, and in some cases perhaps none at all, although I think most physicians always use some. That's what we're trying to achieve. If you look at a normal stem cell, the probability is very, very high that it will have some antigen that's different from the recipient. So if you look at the very first line, an HLA heterozygous donor, which would be an embryonic stem cell, or our heterozygous parthenoids, the club sign would match, but the 0 would not match, and it would be rejected. What happens when we create homozygous parthenoids is illustrated in the second slide. And I'll give you a little bit of biology here that's much simplified, so for those of you who are scientists, please forgive me for oversimplifying. But basically, when a child begins to be born, the first thing that happens is the female throws off half of the DNA that would otherwise be in her egg. That's to make room for the sperm. What happens in parthenogenesis is, and with the homozygous version that we are favoring, is it still throws off that half of the DNA, but then replicates the other half. So you've restored a complete DNA chain, but it matches at each point along that chain. If you think of the DNA helix as a ladder, it matches at every rung of the ladder. That means our chances of matching the recipient go up enormously because we've eliminated one whole side of the DNA that might cause an immune response. The result of that is that we can, in fact, from a single cell line, match almost, in the first case of our first cell lines, about 350 million people. And with a bank of between 50 and 100 cell lines, we can probably match most of the world's population to eliminate or remove immune problems. Put that in context, if you were using embryonic stem cells to do it, the estimates are it would take something like 1,000 lines, and some estimates have been as high as 100,000. So it's enormous potential benefit for us, and it's the reason we're so excited about the parthenogenic stem cells. I'm going to summarize with these next 2 slides briefly by comparing them, the embryonic cells, to what we're doing. Parthenoids, we like to say anything you can do, we can do better. Basically, they will do anything an embryonic stem cell will do. They'll form any cell in the body. The process of getting what they call differentiation is essentially the same. But no fertilized embryos, no life destroyed, and we can deal with immune rejection. And for us, of course, we own the IP. I like to use the analogy that we want to be the intel inside any therapy for human cells. And by that I simply mean we're not interested in which treatment works best. We don't care as long as there is a human treatment that uses stem cells or uses cells derived from stem cells. Our goal is to be the provider of those cells. Similarly, when we compare ourselves to iPS cells, they have the advantage of starting with an adult cell, so they take the ethical issues off the table, but they have a much more complicated path to get back to the point where we actually start, which is the primordial cell. They have to use cancer-causing agents today to do that, and definitely use genetic manipulation. The benefits, personalized medicine, maybe, and a model for studying specific diseases, absolutely. The big disadvantage in the personalized medicine is that ultimately it's one person at a time. It's going to be time-consuming, and it's going to be expensive. By contrast, we're hoping we can provide cells to very large segments of the population at very low cost. The chart that follows is basically puts this in graphical form, and since we drew the chart, why, guess what? Parthenogenic cells turn out to be superior in all of the categories. The reality is, however, that's true. We genuinely believe that they are superior in one way or another to any of the other alternatives, including adult cells. Let me go on then to the question that immediately comes up from almost every scientist that hears about parthenogenic cells for the first time. And that is, can they differentiate into other cell types? And the next few slides are really about that. This slide is a plate showing beating heart cells. Unfortunately, I can't make them beat for purposes of this presentation, but they actually do beat in the Petri dish, which is amazing stuff. But it's typical of the differentiation process that takes place with embryonic stem cells and also with parthenotes. The next slide is one of our wonderful serendipitous discoveries. The little ball in the center of the slide is actually a human cornea, or the human corneal tissue. Contains all of the layers of the cornea, and we believe will be the basis for actually providing cells for transplant for human corneas, and also for doing toxicity testing, which is now done on the eyes of live rabbits, a thing called the Drey's test. We hope to be able to replace that, not only saving the drug companies enormous amounts of money, but saving many, many small animals from what really amounts to a form of torture. So we're very excited about that. The next is a fairly ugly slide showing human pigment cells, but they're important because what are called pigment epithelial cells are the key to treating macular degeneration, and we're in animal trials for that right now. The next slide really answers another question that always comes up to us, and that's, where do you get your stem cell lines? Well, we start with unfertilized embryos from in vitro fertilization clinics. The donors simply do not fertilize all of the eggs. They don't need to fertilize all of them. And we get them from only volunteer donors who are otherwise going through the process, because it's a fairly stressful process on the human body to produce eggs for an IVF procedure. We also do not use any animal byproducts in creating the cell lines, and we comply, of course, with all the rules of international consent. The last item on this slide is what our scientists are most proud of, and that is that every woman who has ever donated cells that became a stem cell line for us also went on to have healthy babies from the eggs that they did fertilize. So we've done nothing that will interfere with their ability to have children. The next question that we get asked is in the next slide, which is, what markets are you targeting? And there is a critical element to our strategy, and what I think is the right strategy in this early stage of development for stem cells. We are only targeting diseases where it is already known that you can either cure a disease or treat a disease using human cells, where the problem is either supply or immune rejection or both. So that leads us to retinal transplants for macular degeneration, one of the leading causes of blindness in adults. We're in animal trials now. If they go as expected, we'll follow that with human trials in the relatively near future. Corneal transplants, we're the only people in the world who've ever grown a human cornea in a Petri dish, and we hope to be able to use those in human trials as well. That's likely to be outside the U.S. simply because in this country there's a very good system for harvesting corneas from donors and implanting them. Outside the U.S. it's a major problem. 1 million people, for example, in India and China are blind from corneal problems. So we will probably address the foreign markets first and then work our way back to the U.S. Liver disease, there's a very desperate need for human liver cells, not so much to treat chronic liver disease, although that would be vastly important, but it is a more difficult target. But initially, the great need is to provide liver cells for people who are on the transplant list. Unlike kidney failure, you can't have dialysis for acute liver failure. If you don't get some sort of transplant rather quickly, you simply die. But it's been known for many years that if you can transplant liver cells, they will restore enough function to keep a patient alive till he can get a whole liver transplant. That's what we're working on initially, and then we'll go on to other things. Diabetes, as I've mentioned earlier, there is a known treatment. It suffers from immune rejection, and it suffers from a lack of cells. So those are the ones we're targeting.